Thanks, Bill. Thanks, inviCRO, for inviting me to present today. It's the first time I've ever done one of these, so I hope it goes well. I'd like to talk to you, as Bill said, about this automated quality control pipeline we built for MRI data in a learning health system. The details of this work can be found in a BioIT World submission. Let's see, is this going to start? Here we go, maybe. I had to press play. The details can be found in the BioIT World submission, an abstract we submitted this year, which I think represents a very strong collaboration between inviCRO and Biogen for maximizing the utility of the IPACs and at the same time giving us complete control and access of our data for downstream analytics. The learning health system is the basis for a demonstration project that we're doing at Biogen called MS PATHS, which stands for Multiple Sclerosis Partners Advancing Technology and Health Solutions. And this project is a collaboration between Biogen and academic healthcare institutions to test the clinical feasibility and utility of collecting highly standardized, high research grade quality data and delivering it across a broad swath of MS patients, a diverse MS patient population, and delivering it to the point of care. The data types we intend to collect in the learning healthcare system range from imaging data, MR imaging data, clinical data from their EMR systems, and standardized biosample collections, so blood samples, and downstream analysis of those blood samples. So the learning health system will ultimately be a repository for about 20,000 patient MRI data sets at the end of next year. The work that went into standardizing each healthcare institution's radiology environments, it involved designing and implementing product sequences, product semen sequences that had very specific acquisition parameters, TR, TE, things like that. And then they were optimized for to be most amenable to our brain volume and T2 lesion volume segmentation algorithms that we're developing at Biogen. So they were very specific. They are very specific and highly standardized, and we needed a way to automatically detect changes in those sequences, those parameters, and then at the same time evaluating image quality for sufficiency in our pipeline, our ultimate analysis pipeline. So it's a pretty straightforward problem, a lot of data to deal with though. This is a really big, complicated looking graph of the pipeline, but what I'd like to just focus on, or to draw your attention to rather, is that it really kind of centers on the IPACS database, the quality control pipeline that is. Centers on the IPACS database, there are a lot of arrows going in and out of it. And then the other key point to make about this is it's modular. It's built in Python. It's modular, and the apart from the querying of the IPACS, the other most important thing to note is the analysis module is all based on mostly free open source software and MATLAB, which many people here probably are familiar with. So if anybody has any questions later, I can refer back to this and give people details as they need them. And I'm just going to start the demo now. Hope it works. Okay, so the first thing that happens is we query the database using the XML DICOM API functionality that the IPACS has. We pull the last time the script was run, the last date it was run, and then query on everything that's new since that last date. And then we download via rsync those data sets only to a local place where we can do analytics on it because it's just easier to do it that way. Then the data dump, we use a Python package called PyDICOM that dumps the metadata. And then the files are converted to NIfTY using MATLAB. And then those NIfTY files are intensity normalized. It's a pre-processing step that's necessary before analyzing any of the MR data. And that's a free source software project called ANTS, Advanced Normalization Tools. Then we just do skull stripping with FSL, which is a very common free software, the brain extraction tool. We run those back into a segmentation routine by ANTS again and extract CSF, white matter, gray matter volumes, binary masks of these volumes, and then run those back into MATLAB and calculate SNR, CNR through traditional mechanisms. And we get a sense for what the SNR, CNR is on the terminal display. But then for the user who, if there's a user who's actually actively monitoring these outputs, there's a display to the screen that highlights clearly any issues or deviations from the protocol, including low SNR, as is the case here. And then the pipeline also runs an automated HTML report and sends me an email, and the site an email. So we're running weekly QA QC on data sets that are coming into us before we integrate with their hospital systems, like their PACS. They actually have to upload the data through a web service that that automatically connects to the IPACS and runs this whole pipeline for us. But then once it's done, it's sent to the site, and someday we'll have this report actually look a little bit nicer, and that might be sent to the site too. And then the last thing to note about this pipeline is that all these these flags that we see, you didn't see any in this particular example, but with the exception of the SNR, those flags are sent back to the IPACS database, and we can see, for example, the SNR value, the brain to CSF CNR value, and the gray matter to white matter CNR value for the T1, and similarly for the flare. And so those are easily queryable at the end of this pipeline. So that's an example of one of the really nice out-of-the-box features of the IPACS, along with other things that we use regularly, like automated ingest of images from our web DICOM uploader. The images you can automatically route them to projects within the IPACS, and those projects can then do automatic things like removing protected health information, which is really important for us. And so all those features make the IPACS a really nice image data management solution for us, but all the back-end access and things that MVicro has really worked hard to give us allowed us to build this whole pipeline in just a matter of weeks, really, with a lot of help from Eli. And the entire pipeline itself has saved us about 91 hours a week versus a manual approach to doing something like this, so 2.25 FTE. So it's a really nice, streamlined kind of thing for our project. And that's really it. I'm sorry it's so short, Bill. I can talk to any of the details if anybody has any questions, but that really kind of summarizes it.
Well, maybe keep it connected and keep your questions on that wonderful, simple slide on the pipeline. I had a question maybe that people in the audience may be curious of is what decisions drove you to, one, look into API, and then once you started investigating the API on the back end, what specific advantages that gave you versus just having data stored on local file share? in terms of accessing that data to fill into your pipeline.
You mean what are the advantages of using the iPACs?
For
Is
I know they made the iPads. But no, the API, you know,
The
gateway.
DICOM XML API?
Yes.
That I really, it's just a lot easier to use that. I feel like the queries I can run are simpler and more one-liners than the MySQL DB Python API that we can call, which would just make simple queries to the database, the SQL database. But I prefer the. I don't know why I actually have a mix of that with some other traditional queries through MySQL DB, but it's definitely seems to be faster and easier to code than anything else. So that's nice.
Any other questions? Maybe from the chat, TJ?
I think you covered it.
Okay.
Thanks, Nicki.
My pleasure.